Hi everyone. In this video, we'll talk about the key types of enzyme-catalyzed metabolic reactions and review some of the key principles of enzyme kinetics that are important to understand metabolism. Okay, so the first thing to remember is that every step of every metabolic pathway is usually catalyzed by a different enzyme. So every arrow on this metabolic map has a name of an enzyme that's making the reaction rate of that process go much, much faster. We have about 1,600 different metabolic enzymes in us, and luckily, you're only expected to know a small number of those. And it's important to remember that the reaction rates and the affinities for all these enzymes and their substrates have evolved to be appropriately balanced to support healthy life. And so if there's any mutation or an enzyme inhibitor that affects these things, that can result in pathology. All right, so the first type of enzyme we'll talk about is a dehydrogenase. Some of these are called reductases in their names instead. These are enzymes that transfer electrons and their hydrogens between different metabolites and the cofactors, either NAD or FAD. So an example of this is lactate dehydrogenase. This is an enzyme that interconverts pyruvate and lactate. And shown here, if pyruvate is the oxidized form of the substrate, lactate dehydrogenase will transfer the electrons and hydrogens from NADH onto pyruvate to make lactate the reduced substrate. And it also takes lactate and NAD plus and oxidizes the lactate to pyruvate, reducing the NAD plus to NADH. So what direction will this reaction go within a cell? And that depends on the exact situation in the cell and usually what the concentration of NADH is in the cell. That's the biggest driver of this reaction. So if NADH levels build up in a cell, which typically happens if the cell is deprived of oxygen or there's some other problem with the mitochondria and the respiratory chain, then cytosolic NADH will build up, and that will push the reaction towards the right here, and lactate will build up. When lactate and a proton are released into the bloodstream, we measure that as lactic acid in the blood, and if that happens to a high level throughout the body, it can result in lactic acidosis. If there are just some cells in the body that become oxygen deprived, so this can happen during vigorous exercise. For example, our muscles, we can push them beyond the oxygen capacity and the muscles will release lactate into the blood. Then cells with plenty of oxygen, such as the liver, can take up the lactate, and in the liver, there'll be plenty of NAD+. Hepatocytes can convert lactate back to pyruvate, producing NADH. The NADH shares its reducing equivalence with the mitochondria and with the respiratory chain, so the NADH gets reoxidized and pyruvate can be used. And in the example we just gave, that pyruvate would largely be used to make glucose via gluconeogenesis. All right, so the next type of reaction is a kinase. Kinase is an enzyme that transfers phosphate, usually from ATP, but sometimes GTP, to a metabolite. An example is hexokinase, making glucose 6-phosphate. And I'll just remind you here that glucokinase is what's called an isozyme of hexokinase. So it is a different enzyme encoded by a different gene that catalyzes the exact same reaction. And it has very different kinetics, has very different affinity for glucose and rate of reaction than hexokinase. And that is really important for maintaining our blood glucose levels. And we'll get back to that at the end of this video when we talk about enzyme kinetics. The third type of enzyme is a phosphatase. Phosphatases hydrolyze a phosphate off of a metabolite. So for example, glucose 6-phosphatase, really important enzyme for releasing glucose primarily from hepatocytes, also from kidney cortex cells during gluconeogenesis and glycogen breakdown to help maintain blood glucose levels. By contrast, a phosphorylase is an enzyme that uses phosphate to cleave a metabolite. And the most well-known phosphorylase is glycogen phosphorylase. So glycogen phosphorylase is an enzyme that activates phosphate, shown here, to release phosphorylated glucose. So here, glucose 1-phosphate comes off of the glycogen. There are other phosphorylases in the body, but this one is so well known that sometimes we just use the word phosphorylase to mean glycogen phosphorylase. And if it's in the muscle, it's often called myophosphorylase. And then just want to remind you about what glycogen is. This shows a two-dimensional version of the structure of glycogen. We have a polymer, highly branched polymer with lots of glucose molecules attached to a central core of protein. And I think what I'd like you to try to do is envision that this, in reality, glycogen is a ball. It's a three-dimensional sphere of glucose molecules connected to each other that need glycogen phosphorylase to release them so that the cell can use the glucose. Okay, so you may have heard of an enzyme called phosphorylase kinase. So what on earth is that? So this is a chance for us to review that enzyme names typically give the substrate first, followed by the type of enzyme second. So the substrate in this case is the enzyme phosphorylase, the enzyme glycogen phosphorylase. And the kind of enzyme is a kinase, a protein kinase. So phosphorylase kinase is a protein kinase that phosphorylates glycogen phosphorylase and regulates its activity. Okay, the fifth type of enzyme we'll talk about is a synthetase. A synthetase attaches a metabolite to something and directly uses the energy from ATP or GTP to catalyze the reaction. So an example of a synthetase is fatty acyl-CoA synthetase. It's an enzyme that takes a fatty acid, a long-chain fatty acid, and attaches it to coenzyme A. Okay, so we get an acyl-CoA. So here is our fatty acid attached to coenzyme A. Okay, there's a similar enzyme called acetyl-CoA synthetase, which takes the 2-carbon little acetate fatty acid and attaches it to coenzyme A to make acetyl-CoA. Okay, so this enzyme, the synthetase, uses ATP, the energy from ATP, in order to synthesize fatty acyl-CoA. And we'll compare that with the synthase, which attaches an activated metabolite to something. So an example of this is glycogen synthase. So glycogen synthase uses UDP glucose, that's the active form of glucose, here's the glucose attached to UDP, and transfers the glucose on to glycogen to increase the size of glycogen. That's glycogen synthase, and UDP is released. Then we have transferases, which transfer part of one metabolite to another. And one transferase you should know about is galactose 1-phosphate uridyl transferase, which is quite a mouthful. So sometimes we just call it by its gene name, GALT. This enzyme takes galactose 1-phosphate, so the phosphorylated form of galactose, and transfers on uridine, the uracil monophosphate here to make UDP galactose and releases glucose 1-phosphate. UDP galactose can now be converted to UDP glucose and further metabolized. So we need this enzyme in order to metabolize galactose. And you should know that classic galactosemia is a metabolic disorder in which this enzyme, galactose 1-phosphate uridyl transferase, the gene encoding it is mutated. Okay, so then we have carboxylases. These are types of synthetases that add a carbon dioxide to a metabolite. And they all require the vitamin cofactor biotin, or vitamin B7. So you should become familiar with pyruvate carboxylase. This is an enzyme that takes pyruvate and adds a CO2 group to make, so we have 3-carbon pyruvate, and we now have, the product is 4-carbon oxaloacetate, which hopefully you recognize from the TCA cycle. Again, ATP is used, the energy of ATP. It's a type of synthetase, and the enzyme, like all carboxylases, uses biotin. This is an important enzyme in gluconeogenesis. So the lactate coming into hepatocytes primarily and converted to pyruvate, then enters gluconeogenesis through pyruvate carboxylase to make oxaloacetate, which then can be converted into glucose. Right, so then there's transaminases, or also called aminotransferases, which transfer amino groups between amino acids and alpha keto acids. These all require pyridoxal phosphate, which we make from vitamin B6. And here's an example you should be familiar with: alanine transaminase, also called ALT. It's one of the liver function tests. This is an enzyme that's in high concentrations in the liver, but also other cell types, including muscle. And severe muscle damage can also cause ALT levels, as well as aspartate transaminase, AST, to increase in the blood. So this is an enzyme that transfers an amino group from glutamate, an amino acid, onto pyruvate, which is an alpha keto acid. So alpha keto meaning the carbon next to a carboxylic acid. Here's the alpha keto group. And we end up making alanine, the amino acid, and the 5-carbon TCA cycled intermediate alpha ketoglutarate. And of course, this reaction can go both directions depending on concentrations of metabolites in the cell. Right, and the final type of enzyme we'll talk about is a lyase. This is a type of enzyme that breaks bonds without activating water to hydrolyze them. So one example is ATP citrate lyase, sometimes also called citrate lyase. And this is an enzyme that breaks cytosolic citrate into oxaloacetate and acetyl-CoA. And it uses ATP to do that, to attach the coenzyme A onto acetyl-CoA. This enzyme you should know as one of the first steps in making fatty acids. So we need to have cytosolic acetyl-CoA to make fatty acids, and ATP citrate lyase gets activated when we synthesize fatty acids. A second example is aldolase, an enzyme in glycolysis and gluconeogenesis. Okay, so here are our list of 10 important types of metabolic enzymes. Hopefully, you will now feel more comfortable when you see the names of these enzymes, knowing what they do. And the name of the enzyme is usually the first name is the substrate or one of the substrates, and the second name tells you the type of enzyme and what kind of reaction is catalyzed. So we'll finish off this video with a reminder about enzyme kinetics and the importance of reaction rates and affinities. All right. So all enzymes have to bind their substrates in order to catalyze a reaction. And because of this, there's two things that matter. One is the affinity of the enzyme and of the substrate, and an approximate measure of that affinity is called the Km, or the Michaelis constant. And the important thing to know is the lower the Km, the higher the affinity, most commonly. And the second thing is that the rate of the reaction when the substrate is bound, which is also called the Vmax. So the higher the Vmax, or maximal velocity, the faster the reaction. So let's look at this by comparing two enzyme isozymes that we've already introduced, hexokinase and glucokinase. So what we'll do is look at the rate of producing phosphorylated glucose compared to the concentration of glucose. And I'm using a scale here of the typical scale for looking at blood glucose concentrations, and the reason for that should become apparent in just a moment. So what we do typically in measuring rates of reaction is by looking at how fast the enzyme catalyzes the reactions at different concentrations of substrate. So I'll show you some examples of that. Okay, so here for hexokinase, the reaction gets going, at a fairly low concentration of glucose, and as we increase the concentration of glucose, we see it increases, but the rate of increase starts slowing down, and even at fairly low concentrations of glucose, begins plateauing. Okay, so if we fit this to the Michaelis-Menten equation, we would get a curve that looks something like this, and from that we could get the Vmax and the Km, and we can approximate it, those values by just looking at the curve. So the maximal rate of reaction is shown approximately here. To get the Km, it's the concentration of glucose at half the maximal rate. So if we go to half the maximal rate, go over to where that intersects with the curve, we see that the Km value for glucose of hexokinase is quite low. Okay, so hexokinase has a relatively low Vmax and a low Km. So let's compare that now to glucokinase. Glucokinase is the isozyme that's expressed both in beta cells and in hepatocytes. So the two types of cells that are really important for managing blood glucose levels. And here we see that at low concentrations of glucose, this is a very sluggish reaction. And in fact, glucokinase has a sigmoidal curve for this reaction as opposed to a hyperbolic curve. And if we fit the curve, we'll see something like this. Fit the data, we'll see a curve that looks something like this. And now on this scale, we don't even max out the rate of the reaction. We have to go to much higher glucose concentrations than shown here. But the half Vmax, we can approximate as I'm showing on this graph. If we drop down to the Km, we see that is greater than the Km for glucose of glucokinase is greater than 100 mg per deciliter. Okay, so why does all this matter? Well, let's think about it in terms of people who inherit mutations in glucokinase that affect the Km or the Vmax. This can give them differing levels of fasting blood glucose compared to typical. So an important thing to remember here is that the rate of insulin secretion depends primarily on the rate at which glucose enters glycolysis in the beta cells, right? So the insulin is secreted from beta cells primarily depending on how much glucose enters those cells. The glucose goes through glycolysis, affects how much ATP is made in those cells, and that stimulates insulin secretion. And the rate at which glucose enters glycolysis in beta cells depends on glucokinase. So I've highlighted, a physiologically relevant range of healthy blood glucose shown in pink here. And we see the rate of the reaction increases dramatically over this range for wild type or normal glucokinase. So what happens if somebody has a mutant that lowers the Km of glucokinase for glucose? Okay. So in that case, the curve will shift, looking something like this. And what we see is in the same range of blood glucose, we have a faster rate of producing glucose 6-phosphate. So we have more glucose that will be trapped and go through glycolysis in those beta cells, resulting in increased insulin secretion. And that will bring the fasting blood glucose to a lower level than normal. If, on the other hand, somebody has a mutant, inherits a mutation that causes a higher Km of glucokinase, that will look something like this. Okay, so the Vmax has not changed, but the Km is greater. And now the reaction occurs more slowly. See, it's slower than the normal enzyme. That will mean that less insulin is secreted and fasting blood glucose will be higher than normal. This is a form of what's called MODY, or maturity onset diabetes of the young. Okay, so let's sort of summarize that. Mutations that lower the Vmax, we didn't specifically talk about those, but if the Vmax is lower, the rate of the reaction will be lower. Or if you have a mutation that increases the Km, then the reaction rate is going to decrease. And this can result in accumulation of substrate or decreased formation of product. For the reaction we just talked about, what was important was the decreased formation of product, resulting in lower amounts of insulin secretion, and that this can cause pathology. In the case of glucokinase, the blood glucose levels could be too high. Okay, so that ends this video. Thank you for watching.